Up next, I would like to introduce Dr. Peter Kang. He is a pediatric neuromuscular neurologist who has seen patients in MDA-affiliated clinics for the past 2 decades. He also runs a research laboratory that studies genetic dilemmas and potential new therapies in muscular dystrophy. Over the years, he has given presentations at local, regional, and national conferences sponsored by the MDA. He recently moved to the University of Minnesota, where he is the director of the Paul and Sheila Wellstone Muscular Dystrophy Center and professor of neurology. So, Dr. Kang, thanks for being with us. I will turn it over to you.
Thank you very much, Nicole, and many thanks to the MDA for this invitation. I will start to share my screen. So today I'll be speaking about clinical studies and treatment trials for limb-girdle muscular dystrophy. And many thanks to Nicole for that nice introduction. So disclosures, I do have a handful of corporate affiliations, and what I'll be doing during the course of this talk is to avoid any mention of specific company names or brand names. So we'll go through a few topics. Under clinical studies, I'll go through genetics of limb-girdle muscular dystrophy, and I know that Stephanie Bavona later on will be giving a really nice talk on genetics. And so I don't expect that my material will overlap too much with hers, but since I do laboratory research on this and I'm actively enrolling participants, I thought I would mention it. Then I'll discuss MDSTARNET, which is a really wonderful consortium that I participate in, and then Grassbell LGMD, another wonderful consortium that I participate in for limb-girdle muscular dystrophy. And then we'll go on to some treatment trials. It is a very exciting time in this field. So for genetics of limb-girdle muscular dystrophy, this is a question that has come up repeatedly, less so now because I think people appreciate more the power of genetics these days. But earlier in my career especially, the question would come up, why should we bother finding the mutation when we see a patient with a genetic disorder like limb-girdle muscular dystrophy? And so I do see this question or hear this question less often now, but even today, once in a while, I do hear it. So I do want to go over that briefly. It is really important to have a genetic diagnosis if you have a disease like limb-girdle muscular dystrophy. It's critical for genetic counseling and family planning purposes, and then also for long-term coverage for supportive therapies. We want to find the mutation for limb-girdle muscular dystrophy in every patient possible because we want to be sure we have the right diagnosis. There are some treatable diseases that can look a lot like limb-girdle muscular dystrophy, and I've seen examples of this in my career. So one example is spinal muscular atrophy type 3, and that has a treatment available. Another is Pompe disease. The later onset form of that can look a lot like limb-girdle muscular dystrophy, so you want to make sure it's not that. And that also has a treatment available. And then there are congenital myasthenic syndromes, and that's another big category of genetic disorders like limb-girdle muscular dystrophy. There are a few congenital myasthenic diseases that look a lot like limb-girdle muscular dystrophy, and there are treatments available for some of those. Another reason why we want to find the mutation is that the natural history and the prognosis may vary depending on the exact diagnosis. So if you know the exact genetic issue that's going on, we can never have a perfect crystal ball, but we can at least give you more of an idea of what to expect in the future potentially. Supportive therapies can impact the outcomes, and for insurance purposes, it is helpful to have the most precise diagnosis possible if you need coverage for specific therapies that are ongoing. And what's become more important lately is that both traditional drugs and molecular therapies target specific genes or even specific mutations. So for therapeutic reasons, it's becoming more and more important to know exactly what's going on with your genetic makeup. So 2016 is a year that really changed the neuromuscular world for the better. And although the two diseases that I'm highlighting on this slide are not limb-girdle muscular dystrophy, they are both neuromuscular diseases of genetic origin. And so I think as a group, we can learn a lot from the successes that we've seen. So starting in 2016, there have been multiple FDA-approved medications for both diseases. For Duchenne, we're up to 5, amazingly enough. And for spinal muscular atrophy, we've got 3. ASO, which you'll see scattered throughout this slide, stands for antisense oligonucleotides, and those are little small bits of nucleic acids that can do things to the pre-mRNA to alter how genes are spliced, et cetera, as they get translated into proteins. So now that we are all on board in terms of the need for genetic testing and genetic diagnosis, what are the options in terms of techniques? So for decades, we've had a test available called the karyotype. And if you look up in the upper right-hand corner, you'll see an image of a karyotype. And that's basically a microscopic photograph taken of chromosomes. And it is an amazing feat, and it's been around for decades. And it's used to diagnose very large problems with the DNA. For example, Down syndrome is a disease that's classically diagnosed with karyotypes. So it's still in some settings a useful test to do, but there are multiple other technologies available that have come on board over the years. So there's another one called Southern blot, another one called PCR, which a lot of you may have heard of, and variants of these tests. There's Sanger sequencing. Before, when I started my career, that was just called sequencing because that was the only kind of DNA sequencing available. But now that there are other forms, people refer to as Sanger sequencing after the scientist who invented that specific technique. And then there are others that I refer to as more newfangled. And these include chromosomal microarrays and next generation sequencing. So next generation sequencing is a broad term. It encompasses targeted sequence capture, which is what, when you send the muscular dystrophy panel from the clinic, that's what you're typically sending, is a targeted sequence capture. Exome sequencing is becoming more and more commonly used in the clinic, and it's very widely accessible, at least in the United States and several other countries. And then there's genome sequencing, which is available clinically, but is mostly done on a research basis at this point. None of these genetic test methods is perfect. In the lower right-hand corner, you'll see an image from a next-generation sequencing run, and you can see these letters all stand for different nucleic acids. There's a lot of overlap, and there's a lot of irregularity. There are lots of fragments of DNA that are sequenced when you do things like this. And what I'm trying to explain with this image is that you need a lot of computational power. You need fairly high-powered computers, and you need personnel who have expertise in handling this type of data to get something meaningful out of this. So that's what sometimes takes a while and sometimes leads to complications in interpretation. This image shows you some of the complexity of what we discovered over the years for muscle fibers. So the top of the image shows the outside of the muscle fiber. The double layer that you see in the middle is the muscle membrane, and those coat the outsides of each muscle fiber. And then the bottom image shows you what the inside of muscle fiber looks like. And this is a schematic diagram, of course. But you can see that there are dozens of proteins that help the muscle function. They're either inside the muscle fiber, in the membrane, or sometimes even outside. And a lot of these proteins are produced by genes that are linked to muscular dystrophies, especially limb-girdle muscular dystrophies. Now, some of you may have heard of this, but the classification system for limb-girdle muscular dystrophies is changing. And not everybody has uniformly accepted the new classification, but it is becoming more widely used. The reason why this came up is that for recessive limb-girdles in particular, they were called type 2. And a lot of you in the audience who have diagnoses probably know your subtype. Some of you may have type 2A or 2B, etc. The problem is we've discovered so many genes we got to 2Z, and we realized that we really didn't have good options for classifying it. And there's the likelihood that there'll be more, maybe another dozen or two more after that. So to make things a little bit simpler moving forward, a group got together a couple of years ago and came up with a new system. So the recessive limb-girdle dystrophies are now called R1, R2, R3, etc. And that, of course, has no limit when you use a number instead of a letter at the end. And then the dominant ones start with D1, D2, and D3. The diagram on the right, the little circle, it shows you what proportion of patients roughly you can expect diagnoses or answers on after different approaches. So when you send genetic testing from the clinic, most of the series that have been published with large groups of patients with limb-girdle indicate that you can get a diagnosis probably about 50% of the time for limb-girdle patients. But what about the other 50%? So what I've been doing in my lab for a number of years now is enrolling patients who can't get a diagnosis after clinical testing. And what we do is a combination of exome sequencing, genome sequencing, and other approaches to try to figure out what's going on with the remainder. And what we've seen over the years that in our group, which are the patients who are not diagnosed in the clinic, we can figure out what's going on in about half of them overall. So that's pretty good also. So collectively, between clinical genetic testing and then research studies such as the ones that we do in my lab, we can get a diagnosis in about 75% of patients. So then that leaves the question, what about the other 25%? And I've seen repeatedly in the course of my career that at least 25% of limb-girdle patients do not have a genetic diagnosis, and it's probably more in some groups. So what have we been doing? So since 2011, we've been running, and that's about when I've started doing a lot of exome sequencing. We've run a lot of exomes, and we've published a number of papers describing the results of exome sequencing for limb-girdle in the United States, in Saudi Arabia, and in Egypt. And we've discovered a lot of patients who've had diagnoses and genes that are known. But recently, we discovered a new muscular dystrophy gene, and some of the patients that we've described look a lot like they have limb-girdle muscular dystrophy. So this looks like a new limb-girdle gene. So this is something that we published just a couple months ago. The new muscular dystrophy gene is called JAG2. And we were able to assemble an international consortium and got together 13 families. A lot of them had multiple affected family members, and they all had mutations in this gene JAG2. This image here shows MRI images from some of the patients in that paper. And you can see the arrows are pointing to fairly distinctive features you can see on muscle MRI. One of the lessons we learned in writing up this paper is how useful muscle MRI can be in some diagnostic settings. Not every patient needs a muscle MRI, but it is non-invasive. And so it's something that can be very helpful in certain situations. So we still have a group of patients, and some of you might be in this audience today who don't have a genetic diagnosis. What are the missing links? And so two of the things we're looking at in my laboratory are splice site mutations. Transcriptome sequencing, which is an RNA sequencing technique, is becoming increasingly popular, and we do that in my laboratory. We've also come up with a computer program to look for splice site mutations. The other thing that we're looking at is structural variants. And we're trying to look at long-read sequencing, which is another form of genome sequencing, to detect some of these mutations more reliably. This is something that's only available on a research basis. And I'm hoping that as we discover more and prove how useful this can be, that it will be adopted by clinical laboratories. So I'll end this section by just mentioning briefly that we do have an ongoing project in my laboratory. As Nicole mentioned, I recently moved to the University of Minnesota, and we're submitting IRB protocols so that we can continue our work. So in the summer of 2021, I expect that we'll begin enrolling new subjects. For anyone in the audience who may have actually enrolled in my study at the University of Florida, we were able to get consents from a lot of those individuals to move samples and information with us. So those of you who've already enrolled, especially if you've reconsented for this, we will be able to continue studying what's going on with your case. And we may have to contact you again for additional paperwork, but that we'll worry about in the summer. So we are focusing a lot on limb-girdle patients who don't have a genetic cause. We are also enrolling patients who do have known diagnoses of limb-girdle for other studies of disease mechanisms. So essentially anybody who has a clear diagnosis of limb-girdle with or without a diagnosis is potentially eligible. You're welcome to contact me at this email address. We have a new genetic counselor who will be starting in a week or two. And once our IRB protocols are in place a little bit later this summer, we can become much more active with that. So, so please contact me maybe a little bit later in the summer, and I and my colleagues will have more information for anyone who's interested. So I'll move on to the next topic, which is MDSTARNET. And this is a consortium that I joined in 2019. MDSTARNET has a long and distinguished history, starting in 2001, which is 20 years ago now, due to the MD CARE Act, which stands for the Muscular Dystrophy Community Assistance, Research, and Education Act. It was reauthorized in 2008 and 2014. And I think it's had a lot of popular support because a lot has been accomplished with this legislation. One of the components of this was the creation of a national muscular dystrophy epidemiology program at the CDC. And so as a result of this, the CDC was able to start funding a national consortium to do epidemiological surveillance for muscular dystrophy. So what is MDSTARNET? It's a multi-site surveillance system across the United States. It focuses on 9 types of muscular dystrophies. There's Duchenne and Becker, myotonic muscular dystrophy, FSHD, OPMD. And then I underlined 4 subtypes that they refer to as the quartet, and one of them is limb-girdle dystrophy. And so it includes Emery-Dreifuss, limb-girdle, congenital, and distal. The objectives are to characterize the prevalence, natural history, health service utilization and costs, and disparities in access to care. And we do want to assess whether specific treatments, interventions, or changes in healthcare use are associated with disease progression and survival. So these studies require either IRB approval or public health authority, and that's something that is negotiated with the Department of Health in the specific state where the study is being done. Now, these studies are done on a population-based basis, and that's important for equity and inclusion because when you have certain clinics, they may see patients only from a certain subset of the population. So to get really good data on what's going on in a segment of the United States, it's really important to be as inclusive as possible. If you're defining a geographic area, try to find every single patient you can in that area. Longitudinal follow-up is also important. And the data sources that are primarily being used are medical records, including electronic medical records. That's what EMR stands for. And primarily these will be physician notes and lab documents from neuromuscular clinics, but they may also be from other sources. Administrative data is also important, and these will include birth and death certificates and hospital discharge data. So as I mentioned, this network has a long history, and what they've been doing is chunks of 5 or 10 years of typically of activity with a little bit of variation. And there are some sites such as Iowa and New York that have been involved since the inception of this project. And other sites have been present for 1 or more phases, and then sometimes they rotate and sometimes they stay. The initial focus was on Duchenne and Becker, and then in 2011, the focus expanded to all eligible muscular dystrophies. So I have a few maps just to show people how the sites have migrated over the years so that we are covering as much of the United States as possible. And so for phase 1, you can see that there's multiple states throughout the country. Phase 2 is very similar. In phase 3, there was a little bit more rotation that started happening. And I think this is important to make sure that we cover as much ground as possible. And then phase 4 is where we're in right now. And this includes multiple sites. I started this site at the University of Florida, which covers 23 counties in northern Florida. And I'm still working with that site after my move to Minnesota. So let's go back to that quartet that I mentioned before. It's abbreviated ELCD. Emery-Dreifuss, limb-girdle, congenital, and distal. Why are these 4 muscular dystrophies grouped together? The main reason is that they have a lot of overlapping clinical features. When you see a patient in clinic with one of those, sometimes it's really hard, and I've encountered this myself in clinic. Sometimes it's really hard to say definitively, is this patient really have Emery-Dreifuss or limb-girdle or congenital muscular dystrophy? And the other reason is that there are overlapping associated genes. There are genetic origins that are common between some of these subtypes. And so there's a question of whether trying to divide them up and treat them separately would create artificial boundaries between these diseases. And so these are lumped together. And these are, each of these subtypes is quite rare. And so we are able to get better numbers for statistical analysis when we look at them as a group. There was surveillance conducted in multiple previous years and then continues during phase 4, which is the current phase of this project. Why do we need epidemiologic data on these 4 muscular dystrophies? Everybody in this audience knows that it's really important that these diseases are recognized and treated as much as possible. But we do want to know who has it and how common it is, and that really helps in terms of planning research and treatment studies. So specific subtypes have been well studied, and for genetic reasons, sometimes you have clusters of a specific subtype in a particular country, and so you might have a significant number of patients with a certain subtype of limb-girdle, and you can describe that pretty well. But broad epidemiologic studies of limb-girdle are sparse. There have been a couple from Italy that have been published over the years, and there's an initial paper from MDSTARNET that was published just this year from an older data set. Right now, we're analyzing data sets from 2014 to 2019, so our efforts in this realm are expanding rapidly. So I'm going to move on now to GRASP limb-girdle muscular dystrophy, which is a consortium that I joined a couple years ago. So the GRASP stands for Genetic Resolution and Assessment Solving Phenotypes in Limb-Girdle Muscular Dystrophy. This is a consortium that was co-founded by Nicholas Johnson at the Virginia Commonwealth University, and it's supported by multiple sources of funding, including foundation, corporate, and NIH sources. So the Grassbell LGMD sites include the following, which you can see on the map. There's the University of Kansas, University of Iowa, Nationwide Children's Hospital in Columbus, Ohio, University of California, Irvine, Washington University. We're starting a new site at the University of Minnesota. The University of Florida site is still active. There is a site at the University of Colorado and Virginia Commonwealth, of course, which is the coordinating site. And there are 2 sites in Europe, Leiden University and Newcastle University. So what the Grassbell LGMD consortium is doing now is what we call natural history studies. And so some of you may be familiar with this term, but others may not. So just to review, what is a natural history study? There are many published studies of clinical features in limb-girdle, but most of them are retrospective. What we mean by retrospective is that we look at existing information, mostly from medical records. And these are very important studies. I've published a number of those myself. So it's not to say that these studies are not useful. They're useful in a specific context. But if we're starting to think more about designing treatment trials and clinical trials for new treatments, we need a different kind of information. And that's where natural history studies come in. Retrospectively, it's really difficult to generate rigorous longitudinal data due to lack of uniformity in clinical settings. So what happens when you look backwards in a medical record, even if a patient's been seen every 6 months for 10 years and you have all those medical records, it might be that different doctors saw that patient in clinic on certain dates, and every doctor has a slightly different style in terms of documenting and assessing. And if they don't use standardized assessment measures, which are typically only used in research settings, then it's really hard to find the same type of data across every visit across however long that time period is. So you need the kind of data that you can plug into statistical analyses, generate graphs, and just get an idea quantitatively, what is the exact progression of a disease over time. So to fill out that need, we need prospective longitudinal data, which means that we enroll a patient, and some of you who participate in these studies understand this. We need someone to enroll, and then we follow them from that point onwards. And we, of course, we're interested in seeing their records, but that's not the main focus of this type of study. And what happens is, instead of coming to a standard doctor's appointment, you'll come in typically to a research center, and a physician and physical therapists and other professionals, nurses, research coordinators will do various assessments, and it's done in a very standardized way so that we collect the same data or the same type of data at most of these visits. And the data are intentionally collected in a way so that they can be plugged into graphs and statistical programs so that we can get a better idea of how these patients really progress over time. And that's the kind of information that's very helpful for a clinical trial, because you need a way to assess whether the intervention or the drug is working. Is it really helping the patients? And so that's a critical question to answer. So getting back to Grassbell LGMD, it's a natural history study platform is one of the things that it does. It's a complex organization, and they do a lot of good work in genetics and clinical trials and natural history studies. One of the big focuses right now is natural history. And so a platform has been developed, and it includes outcome measures that could potentially be included in upcoming clinical trials. The studies are 12 months in duration typically. Of course, that can vary, but that's a standard length. The visits include surveys, muscle strength and function testing, a blood draw, and depending on the exact study, because these are multiple individual studies, there may be a muscle biopsy or muscle MRI involved. So there have been multiple Grassbell LGMD studies, as you can see in the table on the right. And the subtype, and I use both classification systems, the R1, R2 is the new one, and the 2A, 2B is the old one, but they're the same subtype basically. So for example, R1/2A is calpain deficiency. And so you can see that there have been multiple studies that have been ongoing. These are two active studies. I only mentioned the one on the right because it only recently closed in the US, but it was enrolling until fairly recently. And that's the biomarker development study for type R9 FKRP. There's another study that is still open for at least some subtypes for the Grassbell LGMD Consortium, and it's called the Defining Clinical Endpoint Study. And it covers a broad range of subtypes, including R1/2A, which is calpain deficiency. And I'll just mention the gene names because a lot of you are familiar with the genes: dysferlin, the 4 sarcoglycans, anoctamin 5, and DNA JB6. So for anybody who's not familiar with these studies and would like further information, you're welcome to contact Nicholas Johnson at the Virginia Commonwealth University. The project manager, who's the best first point of contact, is Mary St. Romain, and her email is listed here, as well as the phone number for the main office. And so I'll just pause here for a minute in case anyone wants to write this information down. But for anybody around the country who is interested in this project, you can contact this office, and they will refer you to the closest, geographically closest site around the country that might be still enrolling the subjects in question. So one of the things that I want to do with this talk is to make sure the audience is aware of some of the activities going on, and for those who do want to become involved, how you can get involved. So it is important to have some of this contact information. So treatment trials. So this is the last big section, and a lot of people wonder, how do I find clinical trials? Doing a Google search can be helpful, but so much stuff is pulled up on Google, it's really hard to sort through everything. Some of you may be familiar with the clinicaltrials.gov website. It's supported by the NIH. It's a very authoritative site. Just keep in mind that just because something's listed on clinicaltrials.gov, it doesn't mean that there's an endorsement from the government or the NIH. It's just a central repository. But you do have to have a lot of paperwork organized to be listed on clinicaltrials.gov. And so there are certain filters before something is listed there. So these are studies that have cleared certain hurdles, but you have to make individual decisions about whether to participate or not. It's primarily a US study website, but there are international listings on it also. So anyone from around the world should take a look if there are international participants today. So what you can do is go onto this website. There's a box labeled condition or disease. If you just type in limb-girdle muscular dystrophy, you'll pull up everything that is relevant to limb-girdle muscular dystrophy. And when I did this yesterday, I pulled 39 studies. Now, this is also a historical record. There are some studies listed that closed 10 years ago and other studies that are just getting up and started. So you want to look carefully at the information on each listing. And so it does take a little bit of time if you want to go through each one. But this is the clinicaltrials.gov website tries to be as inclusive as possible, assuming that the study has passed the requirements. Now, it's important to also remember not all of these studies are treatment trials. Some of these are natural history studies, like the Grassbell LGMD studies are listed on clinicaltrials.gov. So some of them are more natural history studies and or genetic studies, and not all of these studies are active. But all of the following studies that I'll discuss are listed on clinicaltrials.gov. And again, I do want to repeat, I'm not endorsing a specific study by mentioning it, but these are ones that have recently been posted and recently have been recruiting. Most of them are not actively recruiting right now, but these studies go in multiple phases for groups that want to try to get FDA approval. So if a phase 1 study was enrolling and then closed, there may be a phase 2 study that opens up and in a year or two. So it's worth taking note of what's going on with particular studies. And there may be new ones or new versions of closed studies that open up in the future. So one study that I'll mention, this is a therapeutic candidate for dystrophin deficiency. It makes use of an adeno-associated viral vector, and I'll explain in a few minutes what that is for anyone who's not familiar with that. And it was a study of doing an intramuscular injection into the extensor digitorum brevis, which is a foot muscle. And it was a phase 1 study done at Nationwide Children's Hospital led by Jerry Mendel. And this and an animal study of this approach was published a few years ago. Now, one thing I'd like to address for a minute is the choice of muscle to be injected. For those of you who might be familiar with this muscle, and not everyone will be, but the extensor digitorum brevis is a small muscle in the foot. It helps you wiggle your toes a little bit. And so some of you might ask, why are you testing a therapy in a small muscle in the foot when that's sort of not the big problems that are affecting me? So this is a phase 1 study, and the main goal is to test whether there's safety and tolerability of this intervention. So the reason why this muscle was selected was actually, if you're looking at safety and tolerability, it's better to look at a muscle that is not so consequential in case there are side effects or any problems that come up for your overall function. So that's why this muscle was selected for this study. So you can see a common theme. There are a number of gene therapy studies going on, which is very exciting for limb-girdle. So this is another gene therapy study, and this is a candidate for alpha-sarcoglycan, SGCA, which is limb-girdle type R3/2D. Now this is interesting because it's a study of an intra-arterial limb infusion. So there are multiple ways you can deliver the AAV gene therapy, and unfortunately, every time you try a different approach, you have to start a new study. So there's the intramuscular injection, and you know, even if you target a larger muscle with the intramuscular injection, you probably will need to do multiple injections in multiple muscles. And so then there's the question of feasibility that comes up for administering this. Another way to deliver this is intravascularly or intravenously. And that could potentially go all throughout the body. But there is a concern about dilution of the viral vector throughout the body and not targeting the muscles exactly that you want to. So this other approach that has been studied largely in animals, but here, as you can see, it's also being studied in humans, is to do an intra-arterial limb infusion. So this is a phase 1/2A gene transfer study. It was completed, and it looks like it was published, and this group was led by Jerry Mendel as well. And an intramuscular injection version of this was published about 11 years ago, and then the results of this study were published a couple years ago. This is also listed on clinicaltrials.gov, and this portion of the study is completed. Another treatment is SRP-9003. This one's for beta-sarcoglycan, and this is a third gene therapy candidate. And this is delivered by intravenous infusion, and the study was led by Nationwide Children's Hospital. The status on clinicaltrials.gov is listed as active and not recruiting. So in a situation like this where you're not sure about whether they're accepting your new patients or may in the future, you can always contact the site. There's typically an email address and or a phone number listed, and you can contact them and ask what the status is. If they're not open for enrollment, are they planning future studies? And those are all helpful things to find out if you're interested in participating. And then there is another one. This one is in France, and that was completed a while ago. This is a therapeutic candidate for gamma sarcoglycan using AAV1, which is another version of the AAV virus. This was an intramuscular injection into the carpi radialis muscle, and that's a muscle in the forearm. It was a phase 1 clinical study. It was conducted at the Hospital Pitié-Salpêtrière in Paris, and some information about this was published in 2012. So the last one that I'd like to mention from the clinicaltrials.gov website is BBP418. And this is a candidate for FKRP. BBP418 is a sugar that's a precursor for the FKRP enzyme. So FKRP is a gene, but the gene encodes a protein that serves a particular function in muscle. What the FKRP enzyme does is it attaches sugar molecules to part of the muscle fiber called alpha-dystroglycan. And that's a very important thing that the FKRP enzyme does because it helps with stability of the muscle fiber and muscle function. So this sugar is one that's naturally occurring, but the idea is that if you provide more of this, which is something that the enzyme needs to polish the muscle fiber, that activity will be enhanced and that there'll be therapeutic benefit. One nice thing about this is that it looks like there's oral administration. This study is being conducted at Virginia Commonwealth University. Dr. Johnson is the principal investigator. The status is listed as enrolling by invitation. So to find out more about the status of this in future studies as this compound, I recommend contacting Virginia Commonwealth. So within clinical trials, I thought I'd talk about gene therapy for just a few minutes to wrap up, because gene therapy is in the news a lot. And as you can see, there have been a lot of studies of gene therapy in the past few years. So a traditional gene therapy approach, and I'm actually very happy to use the term traditional gene therapy approach. That indicates that gene therapy has been around for a while. It's been studied for several decades. But the reason why I say traditional is that there's so many different forms of gene therapy that have been developed now that, that it's important to point out what we're talking about. So the traditional approach to gene therapy is if you have a recessive disease, which is what a lot of limb-girdle dystrophies are, it means that the gene expression is either absent or severely lowered. And what you do with gene therapy traditionally is that you introduce a copy of the healthy gene into cells and hope that that will be therapeutic. Years ago, before viruses started being used to deliver these, people tried just injecting naked DNA strands because why do we actually need a vector? Can't we just inject DNA? So people tried importing naked DNA strands into cells. The problem was that that was a very fragile thing. The DNA was better off being protected to a large extent during this process. So it was determined quite a while ago that just injecting DNA is not a good idea. So to achieve stable expression, DNA is administered by a gene delivery vehicle, and today it's typically a virus. So gene therapy has been under development for decades, and it's complex. The concept is straightforward, but the actual implementation involves a lot of different animal experiments in the laboratory and then a lot of human clinical trials, as you can see from what I just reviewed. What you want to accomplish is you want long-term expression of the gene. You don't want to go to all the trouble of putting in the gene therapy and then find out the gene's only active for 5 minutes. You want it to be active for a while. You want it to target the organs and tissues of choice. These viruses, especially the ones we use today, do not trigger immune responses as much as the ones that were used earlier on for gene therapy, so they are a lot less toxic. However, the manufacturing is difficult, and you don't want to put more of this into somebody's body than you absolutely have to. So you want to achieve what we call a minimal effective dose. What is the least that you need to inject to get the effect that you want? And part of that is to target the organs and tissues of choice. So if, for say, you want to treat a brain disease, you want as much of the virus to go to the brain as possible, because if it's soaked up by all of the other organs of the body, that means you have to put more in to get an adequate dose in the brain. And so there have been a lot of studies over multiple decades trying to figure out what's the best way of minimizing the dose. You want to avoid an immune reaction. And in, not all, but some gene therapy approaches, you need to administer some immunosuppressant medications. You also want to pay attention to patient safety issues. That's something that is an overriding concern, and that has become very standardized. So gene delivery vehicles, multiple ones have been tested over the years. There have been retroviruses, lentiviruses, adenoviruses, and the one that's most popular for gene therapy, the traditional form of gene therapy today, is what's called adeno-associated viruses, abbreviated AAV. The funny thing about adeno-associated viruses is that biologically it's not really similar to adenovirus. It was first discovered as a contaminant when people were trying to isolate adenoviruses. And so it was physically located near adenoviruses, but is biologically distinct. Adeno-associated viruses are small, and they tend not to trigger as strong an immune reaction as some other viruses that have been tested. So there's been a lot of exciting developments for using AAV gene therapy in humans, and there are now multiple therapies approved by the FDA, and these are just some examples. Leber's congenital amaurosis, that's been approved by the FDA. There have been 2 cancer treatments using CAR T-cell therapy approved by the FDA. And in the neuromuscular realm, the last few years have been very exciting. There's now an AAV-based gene therapy approved for spinal muscular atrophy, which I know is a different disease from limb-girdle, but it is a neuromuscular disease, so it's very exciting that something has been shown to be helpful. And then for Duchenne muscular dystrophy, there has been a lot of publicity around multiple human clinical trials that are ongoing. And so I'm hoping that in the next few years that we have even more developments that come up that are exciting. And then limb-girdle is catching up fairly quickly. There are multiple human clinical trials, if you've seen earlier in this presentation, that are ongoing or have already been completed, and there are many more that are planned. So definitely stay tuned, because there'll be more exciting news coming out about limb-girdle muscular dystrophy in the years to come. So I'd like to point out some resources for families. The Muscular Dystrophy Association is an amazing organization. As Nicole mentioned, I've been affiliated with them for 20 odd years now, and they're really dedicated to helping families. I'd also like to mention 3 other associations that I've had interactions with that have been very positive: the Speak Foundation, Coalition to Cure Calpain 3, and Jane Foundation. And all of these organizations are very much oriented towards trying to help families. And so there are also numerous others internationally. There are just too many to name here. I just wanted to highlight a handful. So for my research laboratory, I'm very fortunate that it's been thriving with this move. And I've hired several new laboratory personnel, some of whom have started and some of whom will be starting soon. And over the years, a lot of the work that I've done, my lab has done on the genetics of muscular dystrophy, has been done in the laboratory by multiple people who've made really important contributions listed here. The Muscular Dystrophy Center at the University of Minnesota is a wonderful organization. I'm very privileged to be the new director of this organization. And Jim Urbasti and Peter Karachunsky in particular have been immensely helpful. Our administrative director, Jenny Meyer, is a wonderful resource for our families and also for the scientists and physicians at our organization. I've had some longstanding collaborations with investigators at Boston Children's Hospital, Tufts Medical Center, and the Broad Institute. And I do want to also thank the MDSTARNET Consortium and the Grassbell LGMD Consortium, especially Nicholas Johnson for the latter, for really helping me contribute more and more to national efforts to limb-girdle muscular dystrophy care. And these are some of the funding sources that have helped me over the years, including, I'd like to highlight, the MDA has been incredibly helpful. And the Wellstone Muscular Dystrophy Center at the University of Minnesota is a great resource. So thank you very much, and I'll be happy to take some questions.
We do have some questions. Would stem cell therapy benefit help prolong the disease?
Stem cell therapy is very promising. In the 1990s, there were a number of human trials for stem cell therapy for Duchenne muscular dystrophy. Unfortunately, they had negative outcomes, so people sort of dropped that line investigation. Or I shouldn't say dropped it. They went back to animal models and were trying to figure out for quite a while how to optimize it. I think now that a lot of additional studies have been done in animals, we'll probably see more stem cell-based therapies being studied in humans in the years to come. We're probably still in that quiet phase, but I expect that there'll be more. The thing you want to be careful about is that you do want to participate in an organized clinical trial. It's not yet a proven therapy, so you have to be really careful about stem cell therapies. There are potential side effects, and you want to be safe as much as possible.
Okay. What is your thought on gene therapies or CRISPR for limb-girdle? Do you think we have a chance at being approved in the near future for getting something like that?
Well, for gene therapy, I think there have been active clinical trials, and there are more trials planned. So, and there are multiple academic centers and companies that are pursuing gene therapy. So I think that there's a good chance we'll have gene therapy approved for some subtype of limb-girdle in the next few years. I don't know which subtype will be first, and I don't know which institution or company will be the first to get there. But I think it's good that there are so many groups that are interested. And so that is very promising. CRISPR-Cas9 was really a game changer when it was invented or discovered, I'd say. Part of it is a natural mechanism, but the applications are enormous. CRISPR-Cas9 has not been fully studied in humans, and there are potential side effects that could be fairly dangerous. So, so I think the CRISPR-Cas9 is something that has a lot of promise, but that's a little bit more further in the future.
Okay. Okay. Is there any new information for limb-girdle that are caused by Desmond gene mutations, specifically rare forms?
Yeah, that's a relatively rare form. I'm not aware of any specific treatment trials right now, but I think 5 years ago, it would have been hard to predict that there'd be so many trials right now for all these subtypes of limb-girdle. So, for the audience member who asked this, don't lose hope. I don't know of any, but I could be surprised. There could be someone working on something that I'm not aware of that could come out soon. So,
we had someone type in, they're listening from India, and they said they need clinical trials in India.
Yeah, we try to do clinical trials as broadly as possible, and especially for these rare diseases where it's hard to find enough patients to get statistically significant data. It does help to broaden the reach. The problem is every time we go to a different country, we have to work with a different regulatory authority, and the complexity keeps increasing. So that's really one of the things that's a barrier. We want to do everything in compliance with all national authorities. And so, yes, it would be wonderful to expand studies further into countries like India. We know that there are affected patients all over the world. It's just some of these regulatory logistical barriers that make it difficult.
True. Is there any supplement that you would recommend anyone with limb-girdle or any subtypes take?
The most common supplement that I recommend to almost all neuromuscular patients is vitamin D.
Okay.
And
Why is that?
and and the dose of vitamin D should be something discussed with your physician, because over the years I've gotten more comfortable recommending higher doses of vitamin D, but there are doses that endocrinologists recommend that even I'm a little bit hesitant to prescribe. So I think you want to see a doctor who's comfortable and with what dose. And so for the patients I have who have significant bone health issues, I do involve an endocrinologist because they're comfortable with higher doses of vitamin D than I am.
Okay.
So yeah, vitamin D is very helpful. There are a lot of supplements that have been studied, some rigorously, some not. I'm not aware of any evidence that any of the other supplements have been particularly useful for muscular dystrophy. So just be cautious. I know that a lot of these are available over the counter and in supplement stores. But just be careful of doses, because sometimes these supplements can have side effects if you take them at high doses.
Do people with limb-girdle have a vitamin D deficiency?
It depends a lot on the environment. And so if you live in an area where you have a lot of sun exposure, it's a little bit less common. And I've been to several medical centers around the country over the years, and I have noticed in more southern climates that the vitamin D deficiency is a little bit less common, and in northern climates a little bit more so. But everybody's at risk. And even if your vitamin D level is not technically deficient, your bone health in general is a bit at risk.
Okay.
So even for those who have normal vitamin D levels, it's not a bad idea to take at least a baseline level of supplements. I usually only prescribe the higher levels for those who are documented to have deficiency, but it never hurts to take at least a normal recommended dose.
Okay. This person says their daughter was diagnosed with limb-girdle at the age of 7 with a muscle biopsy test. So could it be her correct report, or is there another test required to have a correct diagnosis?
So with a muscle biopsy, you can sometimes get a pretty good idea of which subtype, but you typically can't find the genetic mutation. You can do genetic testing on muscle samples, but that's not always the best way to do genetic testing. So it might be worth, if you have access to genetic testing, to explore having that done as well.
And Stephanie Bavona can talk about that a little bit later too. We've got a genetic counselor that's going to speak this afternoon. So a lot of the questions that we did get are kind of referring to genetics and testing other methods. So we can get into that. What about clinical trials for older adults? It seems like people who are older have a harder time trying to find a clinical trial. Can you speak to that?
Enrollment age ranges can vary. I know that sometimes they often extend at least to 65 years for some studies, but maybe not always beyond that. And one thing I should mention is that for genetic studies such as mine, there's typically no age limit. It's only really the natural history and therapy trials. And so I can't really speak to the decision-making that goes into the age restrictions for any specific trial, but I think people generally try to balance the goals of trying to maximize the eligible patient population with trying to make the group as streamlined as possible. There's definitely not an intention to exclude anybody who's eligible. It's really for trying to streamline the data analysis. And I know that families do get upset when they don't meet enrollment criteria and there's a trial that they're really interested in. So that's a difficult decision that we try to make very carefully.
Is it unusual for a middle-aged person to be diagnosed with limb-girdle?
No, that's fairly common. There are subtypes of limb-girdle where symptoms don't start till adulthood. And patients sometimes in their 40s or 50s who are diagnosed and have only had symptoms for a decade or less. So it definitely happens.
And are there any studies that you know of for limb-girdle 1B or 2B?
2B, there was one that I think completed. I know that there are several groups actively working on 2B. I would suggest just going to that website, clinicaltrials.gov, and also that the listings on those websites do change. So you might want to check in once a month. And also, at the MDA, I do want to say again, the MDA is a wonderful resource. So I know the MDA posts a lot of useful information on their website. So the MDA and clinicaltrials.gov are great resources for that.
Yeah, we have a clinical finder tool on our website. Can individuals, this person's wanting to know, can they view the data collected by natural history studies or by the GRASP?
So there are certain ways that data are made available. So usually for reasons of privacy, you can't release the raw data with individual identifiers. So that level of scrutiny is unfortunately not available. There are several ways that data can become available. One is through publications, and you can definitely see aggregate data, and you can, depending on what the publication includes, you can even see for individual subjects what their responses were and data on individual subjects. Obviously, in publications, these subjects are de-identified. So you don't know the identity, but you can see a fairly granular level. Another thing is on the clinicaltrials.gov website, there is a tab for each study where there are results that can be posted. And results are not always posted for every study at any given moment, but that's another place to look. And then it's becoming more common also that today when a publication comes out, that there's a data availability statement and the authors kind of indicate what data is available and who to contact to get access to data. So I'd say depending on the situation, it may be possible to access some data. And also if you participate in a study, you can always contact the participant, the institution that you enrolled with, and typically they'll share what they're allowed to share and able to share. And when the study's ongoing, they typically can't say much. After the study's closed, especially if there's a specific drug or treatment being studied, that sometimes they have to be really quiet about it. And so it's not intentionally meant to try to hide anything or not be transparent, but there are certain rules and regulations that have to be observed. So just contact the center that you've enrolled in, and I think they'll reveal as much as they're allowed to and able to.
Okay, that's really good. There's a lot more goes into it, I think, than we think of just getting the results from that type of thing. A few more questions. I want to be conscious of our time. Are there clinical trials for recessive genes? I mean, this person says there's a lot of clinical for recessive genes. What about dominant genes? Are there clinical trials for people with dominant?
So I made that allusion to dominant or traditional gene therapy, and the non-traditional and newer forms of gene therapy. There are approaches where you combine gene and stem cell therapy, which are really exciting. And then there are also approaches where you take gene therapy, and instead of putting in a gene that's missing, you actually try to silence a gene that's not supposed to be there or or is expressed too much. So depending on the exact situation, the dominant ones are a little bit trickier to address, but people are studying it. And I know that for one particular dominant type, a collaborator on the GRASP network, Dr. Weil at Washington University, has a particular interest in one dominant form of limb-girdle. So there are people studying that form. And so I think for technical reasons, it's taking a little longer to get to human trials, but I expect that there will be some in the future.
And do we know the cause of limb-girdle?
The limb-girdle, it's a genetic disorder, and there are over 30 different genes that have been described that cause it. And so genetically, we know a lot of the origins for a lot of patients. The mechanism of disease, it seems to be multifactorial. Some of these genes encode proteins that are enzymes, and others are more structural. So there's a lot that maintains both muscle health, and the limb-girdle genes seem to be connected to different aspects of muscle health.
Okay. And the last question we can take for time. This person was wondering, what was the result of the phase 1 trial gene therapy for the limb-girdle 2B that was injected in the foot muscle?
Oh, I was not involved in that study.
Okay.
So what I would recommend is go to the clinicaltrials.gov website and contact the center that did that study. And if it's not published, as I mentioned, they'll reveal as much as they can. Depending on whether or not it's published, they may or may not be able to say a lot at this time.
Okay. Well, we have some more questions that we have that unfortunately we can't get to. We will try to type in some answers to those. And we also have quite a few questions that I will be directing to our genetic counselor a little bit later this afternoon. So we will get to those later. So thank you so much, Dr. Kang, for your time.
Many thanks, Nicole, and to the MDA.